subclones, the resistance can be transferred. And that means within a tumor, there is possible communication between the different clones of cells, from the resistance clones to the sensitive clones. That means they're talking to each other. And one of the ways they could communicate, we're trying to explore, is by means of the transfer of exosomes. And these exosomes are actually some packed materials that usually different cells will also secrete. And these different materials packed inside the exosomes could be transferred or could be taken up by other cell populations. So that means, as I'm going to explain to you, exosomes could be a means of communication between cells, and actually in our case here, it may be transferring or talking to each others and saying and transferring the resistance from more resistant subclones to the sensitive subclones of cells. And you can see the confirmation here, if we transfer the exosomes from one subclones to another, you can see after the transferal, the resistance or the expression of some of the mediators we believe to be mediating the resistance will be much elevated compared to the control population of cells. And you can see here the same confirmation is that after the exosomal transfer, compared with different resistant clones to the sensitive clones compared to the control, where there is either no exosome transfer, or there is exosome transfer but no exosome transfer, but also administration of drugs. It seems that compared to the control, some resistance has been transferred from the resistance clone to the originally sensitive clones of cells. So that's what we believe, is that in a tumor, there is the presence of different subclones of cells that we can clone out and also subject them to treatment. And there may be some communication between the resistance clone and the sensitive clones, and that if that communication is present, it may account for the disease progression of the tumor, even if we have given the appropriate drug according to the presence of the oncogenic driver mutation. So further work is being carried on in my lab, and concerning the biomarkers after the EGFR and ALK, right now you may have heard about this immune